talk about B cells. So we have T cells and B cells. So what's the difference? Well, B cells, there are many subtypes, but in this class I'm only going to cover with, yes, that is the Assassin's Creed logo. So again, B cells, they shuffle their genes that make antibodies. So the things that your antibodies, if you've taken immunology or one of the advanced cell and molecular biology classes, the VDJ recombination, that's what I'm talking about. But again, some of us haven't even had genetics or cell biology yet, so just know that B cells shuffle their genes. So remember, if you've taken even basic biology, DNA makes mRNA, makes proteins. So what happens if you randomize DNA? So if you randomly generate new DNA sequences, you're going to randomly generate new RNA sequences. If you have randomly generated mRNA sequences, you're going to have randomly generated proteins. So what do B cells do? They randomly reshuffle the genes that encode antibody proteins. So they're going to end up with random antibody-like proteins. So B cells have B cell receptors, and at the simplest, it's easy to think of them as antibodies that instead of floating around in your plasma, they're actually embedded inside the cells. So they become receptors because they're stuck within the plasma membrane of naive B cells. So remember that you have why am I showing multiple colors? I'm showing it because they're randomly generated. So they're randomly generated hoping to recognize some antigen from some pathogen. So here we have a bunch of antigens from some pathogen that was neutralized by a macrophage or neutrophil. So now we have all these antigens, but which do we have matches? Well, again, we don't have matches and recognition here, but hey, it looks like we have a match here. So again, if there's no match, these naive B cells don't get activated. But if an antigen binds to a naive B cell receptor, again, these are all randomly generated. But then it's going to slightly activate, it's going to prime it, but it's not going to be fully activated yet. Something else needs to happen. So what does the B cell do? So now it has an antigen that it's born to recognize, and now it's going to actually bring it in and then present it on an MHC class 2 receptor. So again, B cells are professional APCs. So now it's bringing in this antigen that it bound, and now it's displaying it to the other cells in the immune system. So the thing is that B cells, they're very powerful when they become plasma cells, but they actually have a double check system. They don't get activated once they first bind their antigen. They're kind of like cautious. So then how do they double check with another cell? Maybe they want to double check with another cell that recognizes that same antigen. So say we have a dendritic cell. So let's do a little review. So our naive T cells. So remember, naive T cells, not only can they become cytotoxic T cells, they can also become helper T cells. So this T cell receptor, hey, it's recognizing the same antigen, just in a different context. So this T cell also recognizes that same pathogenic protein. So it's going to be a helper T cell. So here we have our B cell that has bound that same antigen in that previous slide. So it's another B cell that recognizes the same antigen as that helper T cell. So this helper T cell is going to say, hey, I saw the same thing too. I think you're right. This antigen, this pathogen really is here. So now this helper T cell is going to give the B cell a license to say, hey, double check your work. The antigen really is here. Now you can finally become a plasma cell. So the B cell gets very happy because now not only is it wasn't sure at first, but now it's like 100% sure. Now it can finally become fully activated. So when a B cell becomes fully activated in terms of like humoral or antibody immunity, they become plasma cells. So remember those B cell receptors that are like antibodies that are stuck in the plasma membrane? Now the B cell, once it becomes a plasma cell, can actually start to secrete this same protein. So instead of being stuck in the cell membrane, now it can secrete in the environment and then start using these antibodies. Now that they've been trained to recognize this pathogen antigen, now they can bind that antigen on any other pathogens that have that same antigen, and then allow the rest of the immune system to help to combat against that pathogen. So again, here we have opsonization. So they recognize antigen, but they can also coat the pathogen, as we saw in the previous example. So this allows the thought, not only can they do opsonization, so this is another reason why, especially with the COVID-19, I'm surprised they haven't they haven't thought of it earlier. But one thing they did with the Ebola patients, when the Ebola infections in Africa, so until they had like vaccines and antiviral drugs, what they try to do is like for people who survived Ebola, they actually, again, nobody is born with the resistance to Ebola, but if they survive it, now they have antibodies against Ebola. So one thing they did is take the plasma from people who survived Ebola and then transfuse them into people who are combating Ebola. So what does that do? Well, now these antibodies that recognize Ebola can attack and coat and opsonize these pathogens and help your immune system, the patient or the recipient who's fighting Ebola, to help their immune system. So they're kind of acting like, this is what you call passive immunity. They're kind of like helping an untrained immune system. They're kind of giving them a head start. So they're actually doing it with COVID too. So for coronavirus or SARS-CoV-2 survivors and COVID survivors. Yep, so that's where they're trying to push through New York. So they're trying to use a similar setting. So now these people who survived it, they probably have antibodies against SARS-CoV-2. So by transfusing their plasma, which hopefully has antibodies against SARS-CoV-2, into people who are combating it, that can hopefully give those people who are fighting and SARS-CoV-2 and COVID, hopefully give them ability to fight that infection. So not only does it do that opsonization coding, it can also allow macrophages to bind to pathogens. So the thing about macrophages, like they actually love to grab onto antibodies. So if you have a bunch of antibodies coding a pathogen, it's kind of like a little like like a handhold for the like a little thing like yeah, like here we have a little fork. So the thing about macrophages, they can grab onto an antibody that's bound to a pathogen, and then they use that to help them eat. It can also recruit a complement, as we saw in the previous slide. And then, so dendritic cells, so to put it all together, how do we go from pathogen infection and how do we go all the way to T cells and B cells?